brain research has taken a groundbreaking turn with the development of a highly sophisticated organoid model, one that captures multiple brain regions along with vascular structures, offering an unprecedented window into early human brain development. But here's where it gets controversial. This isn't just another tissue sample. It's a miniature, integrated brain system that more closely resembles the complexity of a real human brain than ever before. If you're curious about how scientists are pushing the boundaries of neuroscience, keep reading. Leading the charge at Johns Hopkins University, Assistant Professor Annie Kathuria from the Department of Biomedical Engineering explains, most brain organoids you come across in scientific literature are limited to a single region, like the cortex, hindbrain, or midbrain. We've successfully grown what we call a multi-region brain organoid, or MRBO, which integrates several different brain areas into one coherent system. This innovative MRBO combines neural tissues from the cerebral cortex, midbrain, hindbrain, and even vascular components derived from induced pluripotent stem cells, iPSCs. The process starts with growing each regional organoid separately, using specific protocols that guide the differentiation based on manipulating signaling pathways. For example, to develop midbrain and hindbrain regions, researchers expose the developing tissue to BMP4 and WNT pathway modulators, essential signaling molecules that influence brain patterning. Endothelial cells, which form blood vessels, are created under mesoderm-inducing conditions, followed by adding growth factors that promote vascular development. After roughly 2 months in culture, the separate tissues naturally come together, forming a unified organoid that mimics early fetal brain stages. The researchers confirmed that the individual regional identities were preserved, yet they fused into a single integrated structure. Using advanced single-nucleus RNA sequencing, they found clear markers indicating the presence of diverse neural and vascular cell types from all the original regions. A Johns Hopkins news release elaborates. This mini-brain retained an extensive range of neuronal types, resembling those found in a 40-day-old human fetus. Approximately 80% of cell types present at the early stages of brain development in humans could be observed in these lab-grown models. Those significantly smaller, containing around 6 to 7 million neurons compared to tens of billions in adult brains. These organoids are invaluable tools for studying early neurodevelopment. Furthermore, unlike typical endothelial cells that form blood vessels, the vascular part of the MRBO contains a more diverse set of cells, such as pericytes, angiogenic progenitors, and stromal cells, that better imitate real brain vasculature. Interestingly, the study also demonstrates that these vascular components aren't just passive structures. They actively influence brain development by supporting the growth and maintenance of interneuron progenitors, especially in the hindbrain, suggesting a central role in early neural maturation. And there's more. The team tracked neural activity over time, observing increasingly coordinated network behaviors, which hints at functional integration beyond mere structure. Molecular analysis and imaging techniques confirmed not only the presence of mature neurons and synaptic proteins, but also early indicators of blood-brain barrier formation, a critical feature for brain health and disease modeling. While this breakthrough opens many doors, the MRBO system isn't perfect yet. It still lacks a blood supply that's truly perfused, and the connectivity between different brain regions isn't fully developed. Nonetheless, scientists see this as a vital step forward, especially in modeling neurodevelopmental disorders and testing new therapies. Including vascular elements helps in studying diseases where blood vessel problems are intertwined with neural issues, such as certain forms of autism, schizophrenia, or vascular dementia. Since many brain diseases affect multiple parts of the brain, understanding their development early on could unlock better treatment targets and drug discovery options, says Kathuria. These organoids allow us to test how potential drugs influence both neural tissue and vascular components, bringing us a step closer to personalized medicine. Looking ahead, researchers aim to enhance these models by adding other critical cell types, like microglia, the brain's immune cells, and extending culture periods to allow further maturation. The ultimate goal is to create more physiologically realistic models that faithfully preserve the complex interplay of cells in the developing human brain, providing an invaluable platform for discovery and innovation in neuroscience.